Hi, I'm Natasha Kitchin, and today I'll be discussing some of my PhD work on the gut microbiota in fetal alcohol spectrum disorders. As you sit there, you are more bacterial than you are human. For every human cell, there is approximately 1.3 bacterial cells. These microorganisms inhabit our entire body, including our skin, eyes, mouths, and our gut. Some bacteria, also known as commensal bacteria, are simply along for the ride, while others, the symbiotic bacteria, offer a mutually beneficial relationship. These symbiotic bacteria play a crucial role in development and maintenance of the human body. While the microbiome is able to buffer some changes in diet, antibiotic use, stress, and particularly drinking alcohol can greatly affect the microbiome. Fetal alcohol spectrum disorders is an overarching term describing 4 diagnoses along a spectrum of increasing severity consequential to prenatal alcohol exposure. Despite the fact that prenatal alcohol exposure is one of the most preventable causes of birth defects, developmental disorders, and intellectual disability, South Africa has the highest prevalence of FASD worldwide, with the Western Cape prevalence reported to be up to 31%, significantly higher than the global prevalence of 0.77%. FASD therefore constitutes a serious health concern and a significant social and economic burden. Alcohol consumption affects the microbial composition within the gut and has been associated with changes in intestinal permeability. A compromised intestinal barrier can result in translocation of bacteria through permeable points in the normally impenetrable intestinal barrier, allowing bacteria to enter the bloodstream. Once in the bloodstream, these bacteria may be transferred to the fetus through the placenta, thereby influencing initial bacterial colonization of the neonate gut. Neurodevelopment is modulated by the gut microbiome via the gut microbiota brain axis, the bidirectional communication axis between the gut microbiome, central nervous system, and gastrointestinal tract. Initial gut bacterial colonization plays a fundamental role in neurodevelopment. Neurocognitive development in the early postnatal environment is dependent on microbial composition and the corresponding microbial metabolic outputs. An altered maternal gut microbiome may result in abnormal infant gut colonization, which can alter crucial functions of gut microbiota and can increase the risk of developing a neurodevelopmental disorder. Our study therefore aimed to compare the gut microbial composition of pregnant women who have an infant with FASD and those who have a neurotypical infant, as well as to compare the gut microbial composition of infants diagnosed with and without FASD. There were no significant differences in alpha diversity between women who gave birth to an infant diagnosed with or without FASD, both with prenatal alcohol exposure and without. There were no significant differences in beta diversity between women who gave birth to an infant with or without FASD. Of the variables investigated, both trimester and Bristol stool scale had an effect on beta diversity, with an adjusted p-value following Benjamini-Hochberg correction of less than 0.05. As expected, trimester had an effect on beta diversity. During the third trimester of pregnancy, estrogen and progesterone impact gut microbial composition through their effect on bacterial metabolism. Bristol stool scale also had an effect on beta diversity. The Bristol stool scale gives an indication of transition time through the gastrointestinal tract. The maternal gut microbiome was dominated by Prevotella in purple, Bacteroides in orange, Succinovibrio in pink, Bacillobacter in teal, and Faecilibacterium in green. Ruminococcus, a beneficial gut microbe, was lower in women with infants diagnosed with FASD. Ruminococcus has similarly been found to be less abundant in people with inflammatory bowel disease. Alloprevotella was significantly higher in women with infants diagnosed with FASD. Interestingly, a recent study found higher proportions of alloprevotella in male mice subjected to early adversity. There were no significant differences in alpha diversity between infants diagnosed with or without FASD, both with prenatal alcohol exposure and without. There were no significant differences in beta diversity between infants diagnosed with and without FASD. Of the variables investigated, both delivery mode and Bristol stool scale had an effect on beta diversity. As expected, delivery mode had an effect on beta diversity. Due to the exposure of newborns to vaginal microbes, the gut microbial composition of vaginally delivered newborns closely resembles that of their mother's vaginal microbiota. In contrast, the gut microbiome of infants born via C-section is dominated by bacterial species that are found on the skin. Bristol stool scale also had an effect on beta diversity. Bristol stool scale of a 9-month infant is a good reflection of their diet. The infant gut microbiome was dominated by Prevotella in purple, Bacteroides in coral, Bifidobacterium in orange, Bacillibacterium in teal, and Escherichia or Shigella in green. Bifidobacteria are a vital part of infant health as they degrade the healthy sugars in breast milk. Bifidobacteria have beneficial probiotic health effects such as beta-lactose tolerance, the prevention of diarrhea, a reduction in food allergies, as well as the inhibition of the growth of pathogenic organisms. Unexpectedly, bifidobacteria was higher in infants diagnosed with FASD. A lower relative abundance of bifidobacteria has been observed in children with autism spectrum disorders, making this finding unexpected. With the introduction of solid food, the dominance of bifidobacteria diminishes due to an increased abundance of Bacteroides and Prevotella. Prevotella, which breaks down vitamin B9 from plant-based food, was higher in infants diagnosed with FASD, a finding that mirrors findings in individuals diagnosed with autism spectrum disorders in other low and middle income countries. Prevotella readily breaks down mucin, a structural component of mucus which protects the colon. An increased abundance of Prevotella may result in a compromised intestinal barrier, allowing bacteria and their metabolic outputs to enter the bloodstream and influence neurodevelopment. Although still exploratory, our study showed microbial differences between neurotypical infants and infants diagnosed with FASD. Microbial differences may contribute to the neurocognitive deficits characteristic of FASD. Although larger studies are required to validate these associations are promising for microbe-based therapeutic interventions to reduce the extent of neurocognitive deficits and the debilitating symptoms associated with FASD. I would like to thank the following people and institutions. Thank you very much.
Natasha, thank you so much for this nice presentation. We can get some questions now. If you like, you can write your questions in the Q&A, or you can raise your hand and I can give you a permission to speak. And that way you can ask your question. I have a question, actually. I was wondering, you have found some differences between the microbiota of the infants with FASD and without FASD. I was wondering, do you have any theory about any other metabolic molecules involvement in this process? I mean, the difference in the microbiota is the only cause of the disease, or these changes in the microbiota somehow affect some other molecules within the body and they are causing the problem. So do you have any theory or preliminary data for that?
So that's a fantastic question. So at the moment, we know that alcohol results in the disorder, but we don't know how. So that's why we're investigating the gut microbiome. Since we have found some differences, we do definitely want to look into specifically different metabolites that are produced by the microbiome. But unfortunately, at the moment, that's not in the scope of my PhD, but it's definitely something we need to look at. It's not only about what's in there, but what they're doing and what they're producing. So that's definitely work we'd like to look into.
Okay, we have one question from the audience. Now, okay, now I think you can speak right now.
Thank you very much.
Right.
Yeah, so that was a great talk, Natasha. So I wanted to find out if you got metadata based on these kids that are related to, well, based on the patients, actually both mother and potential kids, that are related to things like, were all those kids born, or were there other consequences like maybe preterm birth or some things we don't really, or that you didn't present? I'm just curious if there were some other involved metadata that you kind of left out of the talk.
Yeah, so this forms part of a sub-study from an NIH study, so that we have a wealth of data. Obviously, the basic demographic information, like preterm birth, we have nutrition data, we have all sorts of data. It's just trying to figure out what to include, because otherwise you can go down the rabbit hole for 10 years trying to include everything. But yes, we do have, for example, preterm birth information. They're very few. We do have 4 sets of twins, and all of those were born quite young. One set was born at 30 weeks, and they were less than 1 kilogram, which obviously will make a difference. But we do obviously look into the demographic data. None of it is of huge significance except for birth weight. We do find that the infants that go on to develop FASD when they are born have a lower birth weight, but that is a known trait in infants born with FASD. Yes, we have lots of data, and we'll definitely have to analyze it all.
Okay.
Yeah, thanks.
Yeah, that makes sense. Sure, thanks.
Okay, we have one more question in the Q&A.
All right, so she asks, she's curious, have you also constructed networks? And if so, are there differences in network structures between the groups? We have not been able to do that just yet. At the moment, we're just sort of looking at the microbial composition and differences between the groups, but it's definitely something I'd like to look into. If anyone has any suggestions on how to do that, I would always be appreciated.